Good morning, everyone. This is Trishna Ball. I am an assistant professor at Department of Pharmaceutical Sciences and Technology, Birla Institute of Technology, Mesra, Ranchi. Today I am going to talk about targeted drug delivery systems for cancer therapy via polymeric nanoparticles. In this chapter, we are going to discuss the various types of polymeric nanoparticles that are into applications into various fields, as well as the different methods that are used for the preparation of polymeric nanoparticles, along with the different polymers that are used for the preparation of these nanoparticulate drug delivery systems. Before going deeply into these three categories of the discussion topics that we are going to do in this chapter, I'll tell you a small summarized way of these nanoparticles. Nanoparticles are basically those type of drug delivery systems whose size range is less than 100 nanometer preferably, and they are very good devices for targeting into specific diseases which are mainly like cancer, and nowadays many scientists they are working even on various types of other diseases like leishmaniasis, tuberculosis. They are going for targeting of all these unexplored diseases. Basically, when we talk about targeting, targeted drug delivery systems are those type of delivery systems which exactly locate at site specific site or organ or areas without distributing in other non-target, non-specific sites. So, these type of drug delivery systems basically gives us an advantage that they do not get accumulated at unwanted sites, thereby leading to unwanted toxicity, toxicity when by getting accumulated in those sites. So, going further, so let me first have a recall which we have discussed earlier in the previous class in the theory classes, that these nanoparticulate drug delivery systems or nanosystems are basically there are lots of classification of these nanosystems like carbon nanotubes of size range of 0.5 to 3 nanometer diameter and 20 nanometer to 1000 nanometer in diameter, and they are basically third allotropic crystalline forms of carbon sheets, either single layered or multi-layered carbon nanotubes. They have wide application in terms of electrical properties like semiconducting devices, diodes, etc. And they have very good functionalized enhanced solubility, as well as they easily penetrate into the cytoplasm and into the nucleus. Besides these carbon nanotubes, other type of nanosystems like dendrimers having size less than 10 nanometers, and they are highly branched, nearly monodisperse polymeric systems produced by controlled polymerization process, and they can be utilized for long circulatory control devices of bioactives, targeted delivery of bioactives to macrophages, as well as liver targeting. Besides this, metallic nanoparticles of size range less than 100 nanometer, which are basically like gold or silver colloids. Nowadays, lots of other metallic nanoparticles like copper, zinc, nickel are also utilized for targeting cancer cells. These metallic nanoparticles not only they deliver the drugs at the specific cancer cells, but also they have very good utilization in case of gene delivery, and also they are utilized highly as a highly sensitive diagnostic assay, as well as thermal ablation and radiotherapy enhancement. The nanocrystals quantum dots are another type of nanosystems of size range of 2 to 9.5 nanometer, and they are basically semiconducting materials, and they have very long term multiple color imaging of liver cells. They are utilized in multiple color imaging of liver cells, as well as DNA hybridization, immunoassay, receptor mediated endocytosis, labeling of breast cancer marker HERE2, surface of cancer cells. Also, polymeric micelles of size range 10 to 100 nanometer, they are block amphiphilic copolymer micelles, high drug entrapment, payload biostability, having long biostability, and they are also having long circulatory effect, target specific active and passive drug delivery and diagnostic values. Also, the last type of nanosystems which we are going to discuss in this particular topic, that is polymeric nanoparticles. They have the size range from 10 to 1000 nanometer, and they are basically biodegradable, biocompatible, and offer drug protection. Excellent carrier for controlled and sustained delivery of drugs. Sleeve and surface modified nanoparticles can be utilized for active and passive delivery of bioactives. In the previous class, in the theory class, we have already discussed these nanoparticulate drug delivery system, how they are being characterized by the size, as well as there are various techniques for determination of size, like by use of dynamic light scattering technique or photon correlation spectroscopy, that is PCS, or by scanning electron microscopic method or transmission electron microscopic method or atomic force microscopic method. Also, the surface charge of the system can be determined with the help of zeta potential measurement, as well as the surface hydrophobicity can be determined with the help of contact angle measurements, as well as the drug release study can be studied both in vivo and in vivo. Now, proceeding further regarding the preparation of nanoparticles, basically the selection of appropriate method for the preparation of nanoparticles depends upon the physicochemical character of the polymer as well as the drug to be loaded. I shall repeat, the selection of the appropriate method for the preparation of nanoparticles depends basically on the physiochemical character of the polymer and the drug to be loaded. Nanoparticles can be prepared from a variety of materials like proteins, polysaccharides and synthetic polymers. The selection of the matrix material is mainly dependent on many factors including the antigenicity of the final product, biocompatibility and toxicity, the degree of biodegradability, drug release profile that is being desired, inherent properties of the drug, aqueous solubility as well as the stability of the drug. This aqueous solubility and the stability, that basically it is a component of inherent properties of the drug, size of the nanoparticles that is required for the production and which is and for what particular application the what type of nanoparticle is required, that is also very necessary, as well as the surface characteristics like the charge and the permeability. Now, basically there are three generalized methods for the preparation of nanoparticles, like dispersion of preformed polymers, ionic gelation or co-adsorption of hydrophilic polymers and polymerization of monomers. There are various types of biodegradable nanoparticulate polymeric systems that are utilized for the preparation of these systems, like polylactic acid, that is PLA, polycyanoacrylate, that is PCA, poly-DL lactide co-glycolide, that is PLGA, poly-DL lactide co-glycolide, that is PLGA. Then besides this, the various other biodegradable polymers, natural polymers obtained from natural sources like plants, mucilage, leaves, seeds, mucilage, polysaccharides can be highly utilized. Like I'll give you some examples like chitosan, sodium alginate, pectin. All the combinations, various heat and trial combinations of these polymers can be utilized for the preparation of nanoparticles. So let us proceed. As we know that these nanoparticulate systems are highly utilized for the preparation for the therapy of cancer, the world's most deadliest disease. Not only cancer, other diseases are also being explored for the to check the efficiency of these nanoparticles, like like in case of tuberculosis, in case of leishmaniasis, all these diseases are also being targeted. Then in case of cancer, like the various treatments are available for therapy, like surgery, then radiation therapy, chemotherapy via intravenous routes, as well as immunotherapy is also given. But these above mentioned methods suffers from various disadvantages like high toxicity, poor oral bioavailability, lack of water solubility, low therapeutic indices, as well as consistency in circulation, non-specific distribution of and delivery of anti-cancer drug both to normal as well as cancer cells. Now, this is a nanoparticulate system where the ligand is being attached, this green colored part, that is the ligand, and also like it has the drug entrapped within it, that is the yellow dot that represents the drug. Since this type of systems can be easily recognized by the specific cells or specific tissue having the specific receptor, so this type of systems can be considered as active targeted drug delivery systems. Now, these polymeric nanoparticles are basically submicron sized colloidal particles, and an anti-cancer agent of interest is adsorbed or encapsulated or conjugated either within or onto the surface of the PNP. Now, this targeted drug delivery system basically depends upon the drug loaded within the polymeric nanoparticles, and it is used for the sustained release of the anti-cancer therapeutics to the site-specific targets. The design of this targeted drug delivery system basically depends upon the rationale of polymer basically on the composition of the polymer, solubility of the polymer, crystallinity of the polymer, molecular weight, backbone stability, then hydrophobicity, polydispersity, which is being tailored for a particular cargo, that is the drug. The molecular weight of the drug and the charge of the drug is highly responsible for preparation of these polymeric nanoparticles. These are some of the common methods that is used for preparation of polymeric nanoparticles, like emulsification and solvent evaporation extraction process, nanoprecipitation, supercritical anti-solvent system, and salting out methods. In the next presentation, we will be discussing in details about these methods. Thank you.